Hi everyone, so we can start today's session. I'll share my screen and you guys can let me know if I'm audible and visible and if my slides are visible. Okay. Can one of you unmute and let me know if I'm audible and if my slides are visible? Are my slides visible?
Yes, ma'am.
Okay. Thank you. So, good evening, guys. Today is the fourth live session of this course, Biochemistry by Professor K. Subramaniam, where we basically discuss some of the previous year assignment questions and we try to solve these questions, and we also try to understand the concept behind these questions. Okay, so that it's easier for us to solve similar pattern questions. So we'll basically discuss the questions that we have for today, and in case if you have any doubt, you can write in the chat box, and if I know it, I'll try to answer, or else I'll look into it and let you know in the next few sessions. Okay, so we'll start with the first question for today. A blank bond is formed between two monosaccharides. Anyone of you would like to answer? What kind of bond is formed between two monosaccharides? What is the name? We discussed a lot about these monosaccharides and polysaccharides in one of the previous sessions. Anybody? Okay, but what is the particular name of the bond? Okay, so I understand it's a covalent bond definitely, but there is a particular name that we have for the bonds between two monosaccharides. Okay, so it's basically a glycosidic bond. Okay, so it's definitely a chemical covalent bonds. Okay, so basically there are condensation reactions that happen between a hydroxyl oxygen atom on carbon on one sugar on one monosaccharide and the anomeric form of another monosaccharide that result in the formation of the glycosidic bond. So the glycosidic bonds are created when the hemiacetal of a saccharide or a molecule generated from saccharide reacts with the hydroxyl group of another substance such as alcohol. Okay, so a glycosidic linkage is a chemical bond in the form of a covalent connection that connects a carbohydrate sugar molecule to another group, which might be another carbohydrate or not. For example, cellulose that we find in plants, chitin that is found in the cell wall of chitin that is found in some of the arthropods. Okay, so they can form glycosidic linkages. Okay, so how is it formed? The glycosidic bond basically will form by the condensation reaction between the hydroxyl oxygen atom on carbon on one sugar and the anomeric form of another sugar. So glycosides, what are glycosides? Glycosides are substances in which one or more sugars are linked to non-sugar or sugar molecules by glycosidic linkages. For example, glycogen. Glycogen is energy storage for the animal cells containing many glucose units linked by glycosidic bonds. Okay. So we'll discuss a little bit more, but I want to discuss the next question also. Simultaneously, we'll discuss two of the questions together. So can someone try to answer this question? A dash bond is formed between two amino acids. Again, covalent bond definitely, but what is the name? There is a CONH bond formed. C double bond O and then NH. These two are linked together with a bond. In case of amino acids, what is the name of the bond? Okay, so it's a peptide bond. Peptide bond is formed between two amino acids. So if we discuss a little bit about the chemical foundations, obviously there are different chemical bonds and non-covalent interactions as well. Chemical building blocks of cells, chemical equilibrium, energetics is important. So what the goal for our discussion today is reviewing the chemical and biochemical principles essential to the understanding of cell process at the molecular level. Okay. So if we review covalent bonding, the most abundant elements in cells are hydrogen, oxygen, then carbon, nitrogen, phosphorus and sulfur, right? And the number of covalent bonds formed between these elements is shown in this table that you see over here. Okay, so you can see how many covalent bonds can be formed. So as we know that valency of carbon is 4, so it can basically bond with 4 things at the same time, all single bonds. If it's bonding with one thing with a double bond, then there will be 2 more bonds remaining, correct? So we have to know that oxygen and nitrogen have unshared pairs of electrons in their bonding orbitals. So the most common binding orbitals for carbon will be the sp3, which is a tetrahedral shape, sp2, which is trigonal planar, as shown in these structures, if you can see. So here it is a trigonal planar, otherwise it's a tetrahedral. So these are all ball and stick models of some popular chemical structures such as formaldehyde or methane. Okay, so when 4 different constituents are bonded to the sp3 carbon, like in this case methane, the carbon is asymmetrical. So for different substituents, in methane we have all 4 same hydrogen substituents are there. So that is why we are not talking about this. When 4 different substituents are bonded to the sp3 carbon, the carbon becomes asymmetrical. Okay, so those carbons will be chiral and optically active. This will not be optically active, or because this is similar, same substituents attached to the carbon. Okay, so if we talk about some of the common functional groups and linkages in biomolecules, we have hydroxyl group, it's a functional group, OH group, alcohol. Then we have the acyl group, how we see in triacyl glycerol, the C double bond O R. Then we have the ketone group, C double bond O, carbonyl group basically. Then we have the carboxylic acid or carboxyl group, C double bond O and then O minus. Sulfhydryl group or thiols, what we call. We find this in proteins because there are disulfide linkages in proteins. So SH group. Then we have the NH2 or the NH3, which is the amino groups, amines. Then we have the phosphate groups, okay, which we find in phosphorylated molecules. And then we have the pyrophosphate. So in this case, it's a diphosphate if you see over here. There are some linkages, such as the ester linkage. Okay, so if there's an acid and alcohol reacting, that will be an esterification reaction, CO and then C double bond O. We have ether linkage, then we have amide linkage. This is the peptide bond, CONH. See, so there will be CO and there will be NH at one side. So there will be a amide group, an amide bond, a peptide bond. Now, if we talk about water, so water is a polar solvent, obviously. It readily dissolves polar and ionic compounds but not non-polar hydrocarbons. So water molecules are polar because hydrogen and oxygen atoms have substantially different electronegativities. Correct? That is, electronegativities means differential affinity for electrons. Now because electrons are shared unequally, the OH bonds are dipolar and partial positive and negative charges occur on H and O. And this feature plus the fact that the 2 unshared pairs of electrons are located on the top of these sp3 hybridized molecules, it basically creates a net dipole moment. Okay, you can see. So water dipoles interact well with dissolved polar and ionic solutes. Now if you talk about non-covalent interactions, there are various weak electrical bonds between molecules that are non-covalent. So they can be either ionic, that is electrostatic bonds, that is attraction bonds like positive, negative, all of these. We can have hydrogen bonds and van der Waals interactions. So non-covalent bonds are typically much weaker than covalent bonds because they are basically interactions and their stability is only slightly greater than thermal energy in biological systems. However, non-covalent interactions still play important roles in protein and nucleic acid stabilization because they are collectively strong. Okay, if taken together, collectively they will be strong. So we have to know that the hydrophobic effect that drives molecular interactions but is not non-covalent per se. So if you see, these are non-covalent interactions: thermal energy, van der Waals, ionic hydrogen bonds, and then we go to the covalent bonds or the covalent interactions. Okay. Now ionic interactions can occur between cations and anions, and these bonds are non-directional and strength depends on the distance of separation according to the 1 by r square. R square is the distance of separation, and strength will also depend on the medium, that is the dielectric constant of the medium. Okay, and is less polar than non-polar solvent. Okay, so ionic compounds, that is NaCl, that is Na plus and Cl minus, basically it's an ionic compound. So ionic compounds, that is NaCl, which is basically salt, common salt, they are readily dissolved in water. Correct. Solvation space of water will basically surround the ions in the solution. If you can see, and water molecules will ODM, so that the negative ends of the dipoles will contact the cations and the positive ends will contact the anions. So we have Na and Cl, Na plus and Cl minus. If you can see, these are the red and white ones are your water molecules. So you can see the positive ends, negative ends of the dipole of the water will contact the cations or the positively charged group, that is Na plus, whereas the positive ends will contact the anion, which is the negative ions, that is the Cl minus. So what is the dielectric constant? Basically the dielectric constant of a medium will be the ratio of the permittivity of the substance to permittivity of the free space. Okay, so that is what we mean by dielectric constant. If you talk about hydrogen bonds, for hydrogen bonds are non-covalent interactions. Obviously, they are very weak bonds, and they basically occur between the hydrogen atom of a dipolar molecule, such as water, and the unshared electron pair of another atom, can be oxygen or nitrogen. And these bonds basically represent the primary way in which water molecules will interact with themselves. Okay, so water molecule is polar, we know, so there's a lot of hydrogen bonds between them. So this basically represents how water molecule will interact with themselves or many other types of biomolecules. So H bonds are highly directional in that strength depends on the proper alignment of the interacting atoms, and this directionality confers bonding specificity, as with the Watson-Crick H bonds between the bases of double helical DNA. So if you can see the images below, water, if you see, water will be H2O, correct? One second. So you can see there is a hydrogen bond between H of this water molecule with the oxygen of the neighboring water molecule. So that is how hydrogen bond will be formed. Then if you see the side, the neighboring image for alcohol and water, here also there is hydrogen bond formation. In case of amine water also there is hydrogen bond formation. The peptide group and water also shows hydrogen bond formation, and obviously ester group and water, there might be chances of hydrogen bond formation. Now what are van der Waals interactions? These are also non-covalent bonds. Obviously they are bonds that are fluctuating, induced dipoles within the electron clouds of the interacting molecules. So these bonds can occur between non-polar or polar molecules, and they are extremely dependent on the distance of separation between the molecules and are significant only when the electron clouds of the molecules are just touching. Okay, so this is what we call the covalent radius, and this is what we call the van der Waals radius. Okay, these are two O2 molecules, basically oxygen, and you can see that the van der Waals radius and the covalent radius are shown here. Now what is the hydrophobic effect? The hydrophobic effect refers to the entropy driven aggregation of non-polar molecules in aqueous solution, because aqueous solution will be polar, right? And basically the hydrophobic effect means how there will be aggregation of the non-polar molecules in aqueous solution. Basically that will occur to minimize the ordering of water molecules with which they are in contact. So this is not basically an attractive force, but rather a thermodynamically driven process. If you see, these are cage-like structures formed by water molecules surrounding a non-polar solute, and the hydrophobic effect will drive the formation of the membranes and contribute to the folding of the proteins and the formation of double helical DNA. Okay, so the folding of proteins, basically the hydrophobic, basically the non-polar amino acids will be aggregated towards in the hydrophobic region. They will basically fold inside. They will be the interior of the protein. They will not really interact with the aqueous medium that surrounds the protein. Now, what is molecular complementarity? So, structurally complementary molecules can bind to each other. Okay, if you ask me to talk about an example, I'll talk about the lock and key model of enzyme substrate reaction. Correct. So, the enzyme and the substrate. The enzyme has an active site that is structurally complementary to the substrate. That is the basic concept behind the lock and key model, right? So basically, structurally complementary molecules will bind to each other, and binding often is mediated solely by non-covalent interactions. So the extent of molecular complementarity will set binding affinity. So how strongly the molecules will interact, and the specificity is like which molecules will interact. Specificity basically means how specific, what are the specific molecules that are interacting, and affinity is basically how strongly the molecules are interacting. So both affinity and specificity are very important for molecular function. Intermolecular complementarity will establish the 3D shapes of proteins. So if you can see over here, this is a stable complex between protein A and protein B. There's ionic bond, there's hydrogen bond, there's hydrophobic and van der Waals interactions. This is a stable complex. But this one is not so stable, because you can see compared to this is very less non-covalent interactions. So there's nothing basically to keep the complex stable as such. That is why this one is a not that stable complex. Now we are entering into biological macromolecules. Okay, so biological macromolecules, as I have discussed in the previous sessions, they are basically biological polymers that are assembled from the building block monomer units, and in all these cases the polymerization occurs via condensation reactions in which the water is eliminated. So see, this is an amino acid, this is another amino acid. So they will come together and form a polypeptide, and this bond formed between the two amino acids, if you see CONH, as I was saying, it's a peptide bond. That's the name of the bond. Okay, and we'll get a polymer which is polypeptides. For nucleic acids, when the two nucleotides come together and join, we get a phosphodiester bond. And for monosaccharides, when two monosaccharides come together, there is a condensation reaction. If you see, these will come together and link. The H2O will go out, and this reaction will lead to a glycosidic bond. Okay, the nomenclature is very important. Now, delving into a little bit detail. So, eukaryotic cell membranes are composed of molecules such as phospholipids, cholesterol, and proteins. We have talked about the quasi-fluid structure, right? That was proposed by Singer-Nicolson, where they said that there is a phospholipid bilayer on eukaryotic membranes. Apart from the phospholipids, there is also cholesterol and proteins. There can be integral proteins, there can be peripheral proteins, etc. Now, phospholipid bilayers are non-covalent assemblies of phospholipids. There is no covalent interaction between them. It's all non-covalent. Okay, and the hydrophobic fatty acid chains of the phospholipids aggregate together in the bilayers. The two layers of a bilayer are called the leaflets, and bilayers are mostly impermeable to polar and anionic molecules but allow non-polar compounds to pass through. Okay, so the non-polar compounds will be able to pass through, but the polar and anionic molecules will not be able to pass through. If you see, this is a zoomed in image of one phospholipid. So you have the polar group, okay, and then we have the hydrophilic, that is water-loving, hydro-loving head group, and hydrophobic or the water-fearing fatty acid tails. Okay. Now, biomembranes typically contain other lipids such as sterols and glycolipids and proteins. But this is the basic structure of a phospholipid bilayer or eukaryotic cell membranes. Coming to amino acids, there are 20 standard amino acids that we talked about. They are specified by genetic code and polymerized into proteins by ribosomal translation. So, amino acids will contain an alpha carbon to which typically 4 different substituent groups can be attached. Okay. So, can you name one amino acid where 4 different substituent groups are not attached to the alpha carbon, making it optically inactive? There's only one amino acid like that. Can someone say it's the simplest amino acid? Anyone? Anyone? So what I'm saying is the question is amino acids will contain an alpha carbon, the main carbon, right? To which 4 different amino substituent groups will be attached. Okay, that will, since there are 4 different substituent groups attached to the alpha carbon, it makes it chiral and optically active. However, there is one amino acid which is the simplest amino acid that is optically inactive. Anybody can say the name of the amino acid? Let's think for 1 to 2 minutes. We all know the answer. Anybody? Correct. Yes, that's why I was telling him English. Correct, you're right. Glycine is a simplest amino acid. It has 2 H group substituents on the attached to the alpha carbon, which is why it is not asymmetric and it is not optically active. Every other amino acid there's 4 different substituents on the alpha carbon, making it basically optically active. Okay. Now, what are the 4 different substituent groups attached? That's the alpha amino group, alpha carboxyl group, and the variable R group. R group is a side chain. So the R group region or the side chain region basically determines the quality of the, not quality, the categorization of the amino acid. Will it be polar or non-polar? Is it aromatic or non-aromatic? All of that. So the alpha amino and the alpha carboxyl groups are charged at neutral pH. Now, there are 2 possible configurations for these groups, the D and the L stereoisomers, which are basically mirror images of each other or enantiomers. Now, the standard amino acids have the L configuration, that we have already discussed, that the standard sugars have D configuration, the standard amino acids have L configuration. And amino acids are classified based on the characteristics of their R groups, that is the side chain group. Correct? Now, what are the hydrophobic amino acids? So, with the exception of tyrosine, these amino acids lack polar functional groups within the side chains, and the hydrophobic amino acids often are found in the interior of protein, which is what I was saying. Hydrophobic means water fearing. Correct? So, these amino acids will be in the interior of the protein. They will not be on the outside. They will be folded inside. Makes sense? So, hydrophobic amino acids often are found in the interior of the proteins. The hydroxyl group of tyrosine residues exposed at the protein surface can be phosphorylated in receptors. So, tyrosine, if you see over here, this is a hydroxyl group. So, this hydroxyl group sometimes gets phosphorylated in case if you are studying cell signaling or something. So, some proteins are phosphorylated at different places for the activation system to work, the signaling cascade to start. So what happens? This hydroxyl group of this tyrosine residues that are exposed at the protein surface can be phosphorylated in the receptors, basically in signaling. Now, what are the hydrophilic amino acids? So hydrophilic basically means water loving. Philic means loving. So water loving, hydro loving amino acids. So these amino acids are basically charged or H bonding functional groups, okay, within their side chains, and for this reason they often are found at the surface of the proteins. So the R groups of lysine and arginine, these are the basic amino acids, positively charged amino acids. They are positively charged at neutral pH. Whereas the R groups of aspartate and glutamate are negatively charged. Okay, and the histidine R group may or may not be positively charged, depending on its environment. So what is the pH condition and everything, depending on all of that, histidine group may or may not be positively charged. Okay, and serine and threonine can be covalently modified by phosphorylation or glycosylation. Okay, you see these hydrophilic amino acids, the basic amino acids, the acidic amino acids, and the polar amino acids with uncharged R groups. Okay, so if you see there's OH in all of them, so this is what makes it polar. What are the other amino acids? So these amino acids carry out special functions in proteins. So glycine and choline are often found in the turns within proteins. So we know that when we talk about secondary structure of proteins, we are mostly talking about alpha helix and beta sheet, but there are also some loops, turns, bends. So these two amino acids are mostly relevant in those turns within the proteins. Cysteine can be modified with lipid groups or covalently bound to another cysteine in a disulfide bond. Okay, disulfide bond is a covalent bond, and they can contribute to protein folding stability. So if you remember the slide that I showed in one of the previous sessions, there is a primary sequence of protein polypeptide. They will fold upon each other, and if there is any kind of disulfide linkages in the protein, disulfide bonds basically, then that will add on to enhance the protein folding stability. It will make the complex stabler, more stable basically. Coming to nucleotides. Now, nucleotides are the building blocks of nucleic acids, DNA, RNA, deoxyribonucleic acid, and RNA. So, what happens is that nucleotides are composed of a base, a sugar, and one or more phosphates. So, a nucleoside contains just the base plus the sugar. So, nucleotide minus phosphate group is termed as the nucleoside. Now, the sugar is ribonucleic acid in RNA, and 2-deoxyribonucleic acid in DNA. And the most common bases are the purines, adenines, and guanine, and the pyrimidines, uracil, thymine, and cytosine. Okay, thymine is found in DNA, and uracil is found in RNA. So if you see, this is a ribose molecule. This is the adenine. So this is the phosphate nucleotide. Adenosine 5' monophosphate. These are the ribose, and the difference between the two groups, sugar groups in DNA and RNA. Ribose in RNA, deoxyribose in DNA. Then we have seen the structures of purines and pyrimidines. Now, what are the properties of carbohydrates? Now, most common monosaccharide in cells contain 5, that is pentoses, or 6, that means hexoses carbons. Okay, so monosaccharides are further classified as aldoses or ketoses depending on whether they contain an aldehyde or ketone group. So pentoses and hexoses typically form rings. If you can see over here, this is a pentose. It will form a ring, glucopyranose, and hexose, that is 6 carbons, it will form a ring, glucopyranose. Now, aldohexoses occur in 16 stereoisomeric forms. Now, dimers of monosaccharides are called disaccharides, and polymers are called polysaccharides. Okay, that's self-explanatory. Now, common disaccharides are lactose, that is galactose and glucose, or sucrose, glucose and fructose. And these common polysaccharides basically formed from glucose are glycogen in animals, starches and cellulose in plants, or all of these. And the linkages between the monosaccharide units are called glycosidic bonds. This is the question. The linkage between two monosaccharides are called glycosidic bonds. There is a condensation reaction happening with the H2 being released. In case of proteins, the linkage between two amino acids will be the peptide bond. Now, what are the properties of fatty acids? The fatty acids contain polar carboxylic acid group and a hydrocarbon tail of variable length. It can be 14 to 20 carbons, even more in certain organisms. And fatty acids are further classified as saturated or unsaturated depending on their content of double bonds. Okay, so there are 6 double bonds that will introduce kinks into the tail, and they will perturb packing interactions, they will disturb the packing interactions, and they will increase the fluidity of the membrane lipids. So the carboxyl group is joined to glycerol via ester linkages in triacylglycerols and phosphoglycerides. These are some of the fatty acids that predominate in phospholipids. We talked about phospholipid bilayer and everything. So phospholipids are the most prevalent components within biomembranes. So in phosphoglycerides, 2 fatty acids are linked to glycerol at carbons 1 and 2, and a phosphate and polar group such as choline basically are linked to the carbon 3. The phosphate and the polar group constitute the head group, and the head groups of phospholipids commonly that are found in eukaryotic cells are shown here. You can find in this table. Okay, so this is phosphatidylcholine. The head group will be choline. Then this phosphatidylethaноlamines. The head group will be ethanolamine. Obviously, there will be fatty acid side chains, but the head group will be ethanolamine. For phosphatidylserine, the head group will be serine. For phosphatidylinositol, the head group will be inositol. So this is basically, if we are talking about phosphatidylcholine, this will be the head group. If you can see over here, it's a choline, and then we have a tail, hydrophobic tail or fatty acid chain. Wait 1 second. Sorry, give me a moment. Yeah. Okay. So now, yeah. So that was that. So if we talk more about the glycosidic bond, glycosidic bonds are obviously basically they are in the sugar that you eat, the trunks of trees, the hard exoskeleton of lobsters, and even in your DNA. So they are very important for the structure of all these substances and many others. Okay, life would not exist as we know without glycosidic bonds. So glycosidic bonds are covalent chemical bonds that hold together a glycoside. A glycoside will be simply a ring-shaped sugar molecule that will be attached to another molecule, and the sugar ring may be either a 5-membered ring or a 6-membered ring, and the other molecule can be another sugar. Okay, so basically sucrose, that is the sugar that you put in your tea or you use to make any kind of dessert. It is composed of 2 sugar units, a glucose and a fructose, that is linked together by glycosidic bond. Okay, as I also said, the glycosidic bond formed by the condensation reaction, which means that one water molecule is produced during formation of a glycoside. The reverse reaction, the breakage of a glycosidic bond, is a hydrolysis reaction. So one water molecule is used up in the reverse reaction. Now the condensation reaction that occurs when an alcohol group or OH from a molecule will attack the anomeric carbon of a sugar. The anomeric carbon is the central carbon of a hemiacetal. Okay, and that is the carbon has single bonds to 2 oxygen atoms. So one of the oxygen atoms will be a part of the sugar ring and the other will be an OH group. So when the alcohol attacks the anomeric carbon, the OH group bonded to that carbon is replaced by the O of the alcohol and the H of the alcohol is removed. Okay, so that the result basically will be a glycosidic bond, which is a sugar molecule linked to another molecule via an ether group, ether. And ether is an oxygen atom bonded to 2 carbon atoms. It is relatively unreactive compared to other chemical groups such as alcohols. Okay, so therefore glycosides tend to be more stable than free sugars. Now the question comes, are all glycosidic bonds the same? So all glycosidic bonds are not the same, because glycosidic bonds can either be O-linked or N-linked. Okay, so an alcohol group can attack the anomeric carbon to form an ether. However, a glycosidic bond can also occur if the nitrogen atom of an amine group attacks the anomeric carbon instead. So one example of an N-linked glycosidic bond can be the molecule deoxyadenosine. Okay, that can be one good example of that. Okay, now if you talk about peptide bond also, like that we had in the question. So yes, a peptide bond is a covalent bond that links amino acids together to basically form a protein. An amino acid is composed of an amino group, a carboxyl group, and a central carbon, alpha carbon, and R group, right? So this peptide bond, which is a covalent bond, will link these amino acids together, and it will form due to the dehydration synthesis in a reaction. Okay, so basically it also polymerizes. So it's an anabolic process in which water molecule is removed to link the two monomers or the two amino acids together to form a polymer or polypeptide. So we know that amino acids consist of an amino group and carboxyl functional group, and during polymerization the hydroxyl within the carboxyl group of amino acid 1 and the hydrogen within the amino group from amino acid 2 join together to form water. Okay, and water is removed as a byproduct. The resulting amide bond, called a peptide bond, is carbon to nitrogen covalent bond CONH. So CN, carbon to nitrogen covalent bond, that now links the two amino acids together to form a dipeptide. Okay, now the sequence of the amino acids is critical to the functionality of the protein. So in order for this reaction to take place, ATP is necessary as an energy molecule. And anabolic reactions such as dehydration synthesis require energy to take place. Okay, so obviously monomers link together through a process called dehydration synthesis, in which water is removed as a byproduct. This is a peptide. Okay. The result will be a CN bond to link the two amino acids together to form a dipeptide. A water molecule can be removed as a byproduct. So both peptide and glycosidic bonds are covalent bonds, but glycosidic is basically what happens in case of monosaccharides or sugars, whereas peptide bond happens in case of polypeptides or proteins. Okay. Moving on to the next question for today. The entire collection of small molecules in the cell is known as dash. Anybody? Let us all think for 2 minutes. Then we can start. Resume. Anybody? So what is this set of small molecules called? The entire collection of small molecules. Okay, so I'll move ahead. It is basically called metabolome. Okay, so the entire collection of RNA in our system is called transcriptome, mRNA. So the entire collection of proteins in our system is called proteome. The entire collection of lipids in our system is called lipidome. The same way, the entire collection of small molecules in a cell is known as metabolome. Okay. So what is metabolome and what is metabolomics? So metabolomics is basically the systematic study of unique chemical fingerprints that specific cellular processes can leave behind. Okay. So generally speaking, metabolomics is limited to molecules 1.5 kDa or less. But metabolomics is also referred to as a small molecule profiling. Okay. So what it does is basically it provides represents single snapshots in time. Okay, so the metabolic profile basically represents single snapshots in time, and the metabolome is a dynamic entity, and obviously they change from second to second, right? So it depends on how your cells have been grown, what are the growing conditions, what is the stress that is causing it to change. All of these depend on the cell health and cell mood basically. So the metabolome is a dynamic entity and can change from second to second. More recent efforts focus on real-time metabolomics, which can measure hundreds of metabolites in a living cell in real time. So real-time metabolomics is quite fascinating because this will basically be able to ensure hundreds of metabolites are measured in real time, and at the same time living cell can also be visualized. So what molecules are measured in metabolomics? If you ask, so they can be endogenous, that is produced, the molecules which are produced within the body, like amino acids, lipids, cofactors, nucleotides, sugars, hormones, etc. And they can be exogenous, okay, that are produced by the body, like that are external to the organism, like drugs, toxins, environmental contaminants, pesticides, herbicides, etc. Okay, so this is what endogenous and exogenous metabolomics mean. So this is the genetics view of metabolomics. Everything in the DNA is known as genome. Everything in the RNA is known as transcriptome. Everything in the protein this is known as proteome. And whatever comes under this, sugar, nucleotides, amino acids and lipids, all of these will come under the metabolome. Okay. Now, how to find out the metabolomics? What are the easiest approaches to do that? So we can do untargeted shotgun profiling. Basically, it will provide the opportunity to see everything, whatever is there. Many will be known and other will be unknown. So relative abundances of hundreds of pounds need to be done. Okay, so data analysis is obviously much more difficult. Now, potentially a powerful phenotypic tool is between like need to have difference between the samples for various kind of mapping techniques. Then we can go to targeted or classical analytical biochemistry. It basically can optimize conditions for specific metabolites. This sugar can be it will optimize the condition for specific metabolites. Like they can be quantified and the data analysis is much easier. There can be flavonoids, carotenoids, etc. But the downside is that standards are needed. So number of known compounds is very limited. So there should be obviously some standard that should be there to basically correlate our findings. Okay, yeah, done. Coming to the next question for today. Based on the graph given below, calculate the molar absorptivity of sample X given at a path length of 0.5 centimeter. Anyone? I'll give you guys to solve this question. I'll give you 3 to 4 minutes. You can try. You have the graph in front and you know some of the values also that are given in the question. Now, can you give the path length? Sorry, can you give the molar absorptivity of the material? Anybody? Anybody would like to answer? 0.5. Okay, anybody else? Anybody else? Anybody else? Please try. Please try it once. Anybody? Just look at the graph and try to answer. Okay, so we'll discuss. So the answer will be 0.4. Okay, we will discuss how we came to it. So if we talk a little bit about the introduction, so many solutions will obviously have different distinct colors. Okay, and how do the colors come? Because the solutions, different solutions will absorb or transmit at different wavelengths of visible light. Okay, so the intensity of the color will be somewhat proportional to the solution's concentration. So more concentrated will give you a darker solution. Okay, so if you see the images also here, the different solutions will have different colored images. Okay. Yeah. One second, I'm keeping my laptop. Yeah. Now, if you talk about spectrophotometry, it is basically a quantitative method of studying matter. So, it will basically study the amount of light transmitted or absorbed by matter. Okay, this is the electromagnetic spectrum. Okay, now we talk about the spectrophotometer. Okay, so our basic purpose is learning the Beer-Lambert's law. So we will come to the spectrophotometer. Spectrophotometer's job is that it measures absorbance or transmittance of light as a function of wavelength. So there is a cuvette placed, and we can measure the absorbance or transmittance of light as a function of that. So what is the general procedure? The sample is placed in the cuvette holder, in the cuvette. Then the light of selected, the cuvette is placed inside the cuvette holder in the spectrophotometer. Light of selected wavelength is passed through the sample, and the instrument measures the amount of light absorbed by the sample basically. So there will be a negative control. What is the meaning of a negative control? Compounds in a solution or just the solvent itself that may absorb at the same wavelength as the compound being analyzed. So this can be a little complicated analysis. So negative control will be a reference blank. So it will contain everything found in the sample solution except the substance being analyzed. Okay, so why do we need a blank? Anybody? Can anybody answer why can't we just go and measure our sample? Why do we need a blank? Anybody? Why do we need a blank? Think about it, you'll be able to answer. Please unmute and let me know. Anyone? So negative control or reference blank is needed so as to ensure that whatever absorbance we are monitoring or seeing is only because of the sample that is being analyzed. Okay. For example, you have a dye that is dissolved in a particular solvent and it is giving showing a peak at some wavelength. Now, how do you know it is? Yeah, anybody? Yeah. Do you want to say something? Did I hear wrong? Did somebody want to say something? Okay. So basically, when you are analyzing a sample, you need to ensure that only whatever peak you are getting is only because of the sample being analyzed and not because of the solvent or any other factor that is already present. For that, we need a blank. So blank will basically contain everything found in the sample solution except the sample being analyzed. Okay, and we need to calibrate the absorbance reading as well. Okay, Did you understand? Can someone explain the significance of preparing a reference blank while using it to calibrate the spectrophotometer? Anybody? I just explained it. Has anybody used the weighing machine in lab? The weighing machine, how you can weigh some substances. Anybody? Okay, so how we do in case of weighing machine, or how we in general do in case of spectrophotometer, we need to ensure that whatever peak or whatever value we are getting is only, yes. What happened? Am I not audible? Am I not audible? Excuse me. Yeah, yeah. So I was asking, what is the significance of a reference blank? Why do we need a reference blank? Anybody? I hope you understood the significance of reference blank. If not, please let me know. I'll explain again. Okay, I'm hoping nobody's saying anything, so I'm hoping people have understood the significance of reference blank. So moving on, this is a schematic of how a spectrophotometer will work. There's a light source, obviously there's a monochromator, there's an adjustable aperture. So this is the incident light, which is given by I0, and this is the transmitted light, which is given by I. There's some kind of absorbance at the particular wavelength that we are taking, and then there'll be a transmitted light, which is given by I. Okay, and then basically we'll see an output. Okay, so the monochromator will separate and transmit a particular wavelength of light, and I naught or I0 will be the original intensity of incident light, total light, and I will be the intensity of transmitted light. So there's two ways of reporting this. Transmission will be T I by I naught, or percent transmission will be I by I naught into 100%. Okay, absorbance will be A 2 log percentage of T. Now, how do we choose a wavelength? Okay, so basically we use a wavelength at which most light is absorbed. For example, we are using a new novel dye, okay, that has been synthesized in lab. So if you want to see if it absorbs at a particular wavelength, and we want to see the absorbance value, how do we do it? At first, we take the spectra. We take the entire spectra at different wavelengths, and then we see at what wavelength is it showing the peak. Okay, that is how we find the lambda max. So if you see the graph over here, can you tell me what is the lambda max for this particular compound? Anybody? think about it and let me know. I'll not say. So just look at the graph and tell what is the lambda, what can be the lambda max for this compound. This is the lambda max for this compound. Anyway, coming to the Beer-Lambert's law. So Beer-Lambert's law basically describes the relationship between the absorbance of a solution and its concentration. So the amount of light absorbed is proportional to the concentration of the solution. So if the solution is more concentrated, there'll probably be more absorbance. Okay, that is A equal to ECL or ELC. A is absorbance, E or epsilon is molar absorptivity constant. It basically depends on the combination of the solute and the solvent, and also the wavelength. And then L is the length of path traveled by light, and solution C will be the solution concentration, mole per liter. So what does this tell us? So there is a direct relationship between this is a direct relationship between the absorbance and concentration, right? Okay, so there obviously we need empirical evidence as well. For that, people can prepare solutions of known concentration and analyze them at Vmax, and we can try to figure out the plot of absorbance versus the concentration. Okay, and the plot of absorbance, the concentration should be linear. Now, since A is a linear function of C, the equation will be basically what is the equation of a straight line, y equal to mx plus b, where m is the slope. Okay, so similarly it is A equal to epsilon LC, where the slope of the best fit line will be epsilon L. Okay, so how can we use spectrophotometer and Beer's law to find the concentration of an unknown? Does anybody know this? How we can use this law and spectrophotometer to find the concentration of an unknown solution? Please think about it and try to answer. Anybody? Can you think? Sorry? Sorry, I'm not able to understand. Your voice is tracking. Can you repeat or can you write in the chat box? Based on the, then I could not catch. Write in the chat. Okay, I'm not able to understand what you're saying after based on the, but anyway, I'll explain. So basically, if you want to find the concentration of an unknown, you need to prepare a standard curve. Okay, so you cannot tell the concentration of an unknown just like that. You need to have a standard curve that will help you basically correlate the absorbance values with known concentrations of solutions. For example, we are talking about BSA or bovine serum albumin. Okay, now BSA is dissolved in a solvent and given to us. We don't know the concentration, what mg per ml it is, but someone tells us that find out the concentration of this unknown solution. We can do that via absorbance, but for that we need to know what are the absorbance values that are supposed to come for some known concentrations of BSA. Okay, so that is what is called a standard curve. So we'll basically prepare a standardized plot. We'll prepare a plot of A, absorbance, versus C, concentration, for standard known solutions to determine, this will determine the equation. Okay, and then what we'll do, we'll measure the absorbance of the unknown solution. Okay, and then we can use the measurement and the equation to extrapolate and solve for the concentration of the unknown. Okay, for example, this example, so you use the plot to determine the concentration of a solution that is an absorbance of A equal to 0.25. Okay, so this is the standard curve that has been given to us. We know that at a concentration of this, the absorbance value is this. At a concentration of this, the absorption value is this. At a concentration of 0.008, the absorption value is this. At a concentration of this, the absorbance value is this. This is how we know. Okay, now someone has given us a solution which whose absorbance value is 0.025. This part, the blue line that you can see. So once we prepare the standard curve, we'll find out the absorbance value that comes 0.25, it will come over here. Then we'll basically drop a perpendicular on the standard curve and another perpendicular from this place to x-axis, and this will basically give us the concentration of the unknown solution. Okay, we can put everything in the equation. So for this standard curve, this is our equation. Okay, we can put 0.25 as y and then solve for x. Okay, another method can be proportionality, where the BS law tells us that the absorption is proportional to concentrations. Therefore, A1 by A2 equal to C1 by C2. Okay, so if people say they calculate the concentration of a solution COCl2 cobalt chloride that has an absorbance value of 0.4 at lambda max, if 5 into 10 to the power minus 3 mole per liter solution of COCl2 has an absorption of 0.175 under the same conditions. So you can do this. You can do A1 by A2. A1 can be your 0.400, A2 can be your 0.175, A1 can be your C1 can be your 5 into 10 to the power minus 3, and C2 is what you are solving for, what you need to know. So the answer comes C2 equal to 0.0114 mole per liter. Other things that we can do is use the Beer-Law's equation, Beer-Lambert's equation. Okay, so this seems very simple because this is an equation, we just have to put the values and solve. But it is not that simple because sometimes the extinction coefficient, molar absorptivity constant, this value is not known properly. Okay, so if we have a substance, like for example, we want to the molar absorptivity of ethanol in hexane at its lambda max is this. So we have it's given to us. Now calculate the concentration of ethanol in a solution that has an absorbance of 0.652 with a path length of 1.2. So we put everything in the equation and we solve for C, the concentration. So coming to the summary of whatever we discussed, matter interacts with light by absorbing and transmitting different wavelengths, and the specific nature of the interaction depends on the compound's molecular structure. For spectrophotometry, it basically quantitatively describes either the transmittance or the absorbance of a light by a substance. Correct? So the optimal light wavelength to use for analysis is denoted by the lambda max. How do we find out the lambda max? Whenever we are using an unknown solute in a solvent, we basically take the entire spectrum. That is what helps us in finding the lambda max. Okay, and that is basically the wavelength at which maximal light absorbance occurs for that particular sample. So Beer's law states that a substance's absorbance value is directly proportional to its concentration. So if absorbance can be measured, it can be used to determine the concentration of an unknown solution. Okay, so you can familiarize yourself with the three methods by which Beer-Lambert's law can be used to determine the concentration of an unknown solution. Now some useful terminology. For example, colorimetry is the use of human eye to determine the concentration of a colored species. Spectrophotometry is the use of instruments to make the same measurements. So it extends the range of possible measurements beyond those that can be determined by the eye alone. So for colorimetry, there will be visual observation. Because colorimetry is based on inspection of materials with the human eye, it is necessary to review aspects of the visible light. So visible light is a narrow range of electromagnetic waves with a wavelength of 400 to 700. Okay, this is the electromagnetic spectrum. We have the visible light over here. It's very small. Okay, it's only a very small region of the electromagnetic spectrum. Now frequency and energy will be directly proportional, whereas frequency and wavelength will be inversely proportional. See electromagnetic spectrum details. The different types of radiations, what are the frequency ranges for them, what are the wavelength ranges for them, what are the type of transition that's prevalent. Electromagnetic radiation can be characterized by this equation: E, which is energy, H, which is Planck's constant, nu, which is frequency. That is hc by lambda. What is c? C is the speed of light. Lambda is the wavelength. So longer wavelength will have lower energy and shorter wavelength will have higher energy. Now if you talk about the color wheel, VIBGYOR, basically VIBGOR, YOR. So complementary colors lie across the diameter on the color wheel and combine to form white light. Okay, so the color of a compound seen by the eye is the complement of the color of the light absorbed by the colored compound. Right? Thus it completes the color. So if you are observing the color of a compound which is green, the color of light absorbed will be red. Okay, so this is approximate wavelength of the light absorbed. For visual colorimetry, for light shining through a colored solution, the observed intensity of the color is found to be dependent on both the thickness of the absorbing layer, which is the path length, and the concentration of the colored species. Okay, so for one color, if there is a series of solutions of single color, that will demonstrate the effect of either concentration or path length depending on how it is viewed. Okay, so for more than one color, the ratio of an unknown mixture can also be determined by matching the shade of the color to those produced by the from known ratios. So for example, in this example, the ratio of a mixture of red and blue can be determined visibly by comparing the mixture to purples produced from known ratios of red and blue. Okay. So when the product of the concentration and the path length of any two solutions of a colored compound are the same intensity or darkness of the color is observed. So now we have learned a little bit about spectrophotometer. It is basically an instrument that measures the amount of light absorbed or the intensity of color at a given wavelength. We know about the intensity of color that can be given a numerical value by comparing the amount of light prior to passing it through the sample and after passing it through the sample. Okay, so these quantitative measurements of light absorbed are the transmittance and the absorbance. So transmittance is given by this equation: T I by I naught. I naught is the intensity of the incident light, the total light that was incident on that. I is the transmitted light. Okay, so basically I is the intensity of the light after it has gone from one side to another, so transmitted light to the sample, and I naught is the initial light intensity. And absorbance is related to the percentage transmission by this formula. Okay, so this is the incident light, this is the transmitted light, and this solution is absorbing some of the light, and this will be the path length V. So the Beer-Lambert's law is a linear relationship between the absorbance and the concentration of an absorbing species. Okay. We have talked about working concentration and how everything is linked: absorbance and concentration. So if we talk about the simple schematic of a spectrophotometer, we have the lamp that will emit all color of light, that is white light. We have the monochromata which selects only one wavelength, and that wavelength is sent to the sample. And the detector, which is basically detects the wavelength of light that has passed through the sample. So and we also have an amplifier that will increase the signal so that it is easier to read against the background noise. Coming back to the question that we started with today, we have been given this curve. Okay, so and we'll put it to the Beer-Lambert's equation. So according to the curve, when concentration is 1 molar, absorbance is 0.2. When concentration is 2 molar, absorbance is almost 0.4. When concentration is 3 molar, absorbance is almost 0.6. When concentration is 4 molar, absorbance is almost 0.8. And when concentration is 5 molar, the absorbance is almost 1. So from all these values, or putting everything into the equation, we can find out the molar absorptivity, which is 0.4 and not 0.5. It's 0.4. Anybody has any question regarding this? We can discuss. Okay, so the answer will be 0.4. Anybody has any question? Okay, then we'll move to the next question for today. Which of the following is true? There are 5 statements. The biomolecules present in E. coli are different from those present in humans. The role of nucleic acids is to act as reserve energy sources for the cell. Stereoisomers that are not mirror images are called enantiomers. The huge diversity of living organisms are created from relatively fewer metabolites, and glucose has a 6 carbon molecule and is a ketose. Please try and answer this. Which of the following is true, do you think? Please, somebody, please try which of the following is true and which is not true. You guys can take a few minutes and then we'll start discussing. Sorry? Can you write in the chat box, please? I don't know, your voice is cracking. I don't know if it's my network issue. Can you write in the chat box which of the following is true? I'll see. Anybody else? Please, others also try. Okay. Third one is true. Okay, let me see. Third one is the stereoisomers that are not mirror images are enantiomers. Third one is true. Okay. Anybody else? Anybody else having different or same answer? Enantiomers are non-superimposable mirror images. Okay. So let's discuss the answer then. So these two statements are true. The biomolecules present in E. coli are different from those present in humans. This statement is true. Obviously, that's a bacteria and that's a prokaryotic cell, and we have an eukaryotic. Humans are eukaryotic. That's true. The huge diversity of living organisms are created from relatively fewer metabolites. That is also true. Okay. So basically, what are biological molecules? So out of the 92 naturally occurring elements, 16 are known to be important constituents of the living systems. So the important elements are carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, sodium, chlorine, magnesium, iron, copper, iodine, molybdenum, and zinc. Okay, these are the important elements. Out of these, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are the most important elements. Everything that is there in our system is made up of this. Correct? Anybody has any other opinion? No. So if we compare the dry mass, you can see by dry mass how what kind of ratios these elements have. Correct? So all important biological molecules contain carbon, and all molecules that contain carbon are called organic, except for CO2. Okay. Now carbon forms 4 covalent bonds. So this will allow carbon to form molecules with many different shapes. So most biological molecules have a core made of carbon and hydrogen, and molecules can differ in structure and function, and in part because of the different functional groups. For example, we have hydroxyl group. One second. One second. We have hydroxyl group with structural formula of OH. Okay. We have carbonyl group, that is C double bond O. Okay. Like then we have a carboxyl group. We have an amino group, sulfhydryl group, phosphate group, methyl group, and these are the examples. So the major classes of biological molecules that are important for all living things are carbohydrates, lipids, proteins, and nucleic acids. And large biological molecules are called macromolecules. Okay, for example, proteins, you have a functional aspect and structural aspect. So for proteins, amino acids are the monomers, correct? They can be helping us in catalysis, transport, support, etc. Functional aspects will be catalysis and transport. Structural will be catalysis and transport, and structural will be support. Okay, so like for functional aspect, we have the hemoglobin, which is a protein which helps in some kind of function. Structural will be hair, silk. These help in giving structural support. For nucleic acids, we have DNA and RNA. DNA will be the most stable nucleic acid. It is basically what carries the information for the next generation. It encodes the genes, the information about the particular organism, and they can be found in chromosomes. RNA is, however, not stable enough to carry genetic information, but RNA is still needed for gene expression, like for messenger RNA. Like how we know in the central dogma, DNA gets transcribed to mRNA, and then mRNA gets translated into proteins, like that. For lipids, fats and phospholipids, we have glycerol and 3 fatty acids, glycerol, 2 fatty acids, phosphate and polar R groups. So they can be helpful in energy storage, constituents of the cellular membranes, and all of that. We have prostaglandins, which are basically 5 carbon rings with 2 nonpolar tails. They can be used as chemical messengers, like hormones basically. We have steroids, 4 fused carbon rings. We have terpenes, long carbon chains. Now we have carbohydrates, starch, glycogen, cellulose, chitin. So these, for the carbohydrates, the subunit will be our monosaccharide, glucose. Okay, they will be helpful in energy storage, cell wall, structural support. Now macromolecules are built by combining smaller building blocks into polymers. So polysaccharides, which are large carbohydrates, are polymers of the monosaccharides. Okay. These are the monosaccharides. The starch is formed from the monosaccharides. We have discussed about starch previously as well. Now, if you talk about fatty acids, fats are built from fatty acids and glycerol. Okay, the fatty acid can give rise to fat molecule that will be in the adipose cells with fat droplets. Now, proteins are polypeptides, that are polymers of amino acids. Okay, and nucleic acids are polymers of nucleotides. Now, what are carbohydrates? Carbohydrates are basically compounds made up of C, H, and O in the ratio. Okay, examples can be sugar, starch, cellulose, chitin, whatever. And carbohydrates are basically energy reserves. So there was an option that said that nucleic acids are energy reserves. That's untrue, because nucleic acids are only responsible for carrying genetic information and for for converting genetic information to another thing. That's what the nucleic acids are needed for. Carbohydrates are basically used for energy storage and structure. Okay. So if we talk about calories, carbohydrates have a calorie value of 4.1 kilocalories per gram. Now, what is calorie? Calorie is the amount of heat required to raise 1 gram of water 1 degree Celsius. So, kilocalorie is basically the amount of heat required to raise 1 kilogram of water 1 degree Celsius, or 1 gram of water 1000 degrees Celsius. So, calorie is a dietary unit of energy equal to kilocalorie, similar to a kilocalorie. So, 1 gram of sugar has 4.1 calories. Okay. Now, the simplest class of carbohydrates are monosaccharides. They are simple sugars. They'll have 3 to 6 carbons. 3 carbon sugars or the triose can be glyceraldehyde. 5 carbon sugars of the ribose and the deoxyribose. 6 carbon sugars we have glucose, fructose, and galactose. Now, simple sugars exist in different forms and can be drawn in different ways. Okay, so they can be, we discussed about Fisher projection and Howard projection last day. We'll not get into too much detail in today's class, but basically simple sugars can exist in different forms and they can also be drawn in different ways, as you can see from the structure over here. So this is a normal way of writing glucose. Then we have the ring form. Okay, then we basically this ring form. This is we have the chair and boat form as well. There are different ways of writing simple sugars. Now, the 6 carbon sugars all have same chemical formula. Okay, this is H12O6 for glucose. Now, they differ in the placement of the functional groups. Okay, so in some cases OH is over here, in some cases H is over here. So, isomers have the same chemical formula. However, stereoisomers differ in the arrangement of functional group at a single carbon. Okay, so if we talk about glucose and fructose, these are structural isomers. Okay, because the OHH, the difference in the OHH thing is not different, it's same. We have 3OH this side, 3OH this side, 2OH, 3H this side, 3H this side. But however, glucose is an aldehyde. See, C double bond O and H. But fructose is a ketone. So this is structural. Functional group difference is there. So it's a structural isomer. However, glucose and galactose are stereoisomers. Both are aldehydes. If you see CHO, CHO. But if you see, there's an H on the 4th carbon this side, but there's an OH on the 4th carbon this side. So that makes these stereoisomers. Now, monosaccharides are the building blocks of more complex carbohydrates. This is a combination of 2 monosaccharides. Glucose and fructose will come together in the condensation reaction, giving out water, forming sucrose. Galactose and glucose will come together, forming lactose. Now, disaccharides are formed by dehydration synthesis. So, glucose plus fructose, sucrose, glucose plus glucose, maltose. Now, many of the larger biological molecules are built by combining smaller molecules, okay, through dehydration synthesis reactions. And the same molecules can be broken apart into their component building blocks by reactions. Now, polysaccharides are polymers of monosaccharides. And polymers are long chain molecules with repeating subunits. And starches are polymers of glucose. Starches are used basically as energy reserves. They can be either amylose, which are unbranched, if you see over here, or amylopectin, which is branched. Now, each glucose molecule is an energy-rich molecule. So, amylose is an unbranched chain and is produced by plants. Amylopectin is a branched chain and is also produced by plants. Whereas glycogen is highly branched and is produced by animals. Now, polymers of two different isomers of glucose produce polymers with different properties. For example, starch is a polymer of alpha glucose and readily digestible. However, cellulose is a polymer of beta glucose and is indigestible. Cellulose is a structural polysaccharide and used for the construction of cell walls in plants. So, this is the alpha glucose, this is the beta glucose. Correct? If you see in the position of the anomeric carbon, the OH is here down. In case of alpha glucose, in case of beta glucose, the OH is up. That's the only difference. That makes this one makes a starch, this one makes a cellulose. So alpha glucose can be digested, but beta glucose cannot be digested in humans. Another important structural polysaccharide is chitin. Chitin is a polymer of beta glucose molecules that have a nitrogen containing functional group. Okay, so chitin is also not digestible. It is basically found in the cell walls in fungi and to build exoskeletons in some arthropods. So even though chitin does not strictly follow the 1C2H and 1,0 rule for carbohydrate, it is still classified as a structural polysaccharide. Now coming to lipids. Lipids are chemically diverse class of organic molecules that are grouped together because they are all largely non-polar. Correct? And they are non-polar because they are non-polar. They are soluble in organic solvents like chloroform, benzene, acetone, paint thinner, etc. But they are not really soluble in water. The major classes of lipids are fatty acids, neutral fats, phospholipids, steroids, etc. And lipids have many functions, including energy storage, cell membrane structure, vitamins and hormones. They are like substances produced in one part of the body that have effects on other parts of the body. Now, fatty acids are long chain carbon molecules with many carbon hydrogen bonds and a carboxyl group on one end. The carboxyl group can then ionize, releasing an H+. They can come in two varieties, saturated and unsaturated. Saturated fatty acids have no double bonds. All carbons in the chain are bonded to as many hydrogens as possible. Okay, so there will be no saturation in case of saturated fatty acids. There will be all single bonds. No CC double bond. So this is a saturated fatty acid. If you see, there is only one double bond, but between C and O, there is no double bond between the carbons. However, unsaturated fatty acids have some C double bonds also in the chain, and the carbon chain is not completely saturated with hydrogens. That's the difference between saturated fatty acids and unsaturated fatty acids. Now, fatty acids are component of two other classes of lipids, neutral fats and phospholipids. Neutral fats are composed of molecules composed of glycerol with the sugar alcohol. Okay, and these fatty acids. And neutral fats are used for long-term energy storage. Now, neutral fats are also called saturated fats because if their fatty acid chains are saturated, that is, if there is no double bond between different carbons. They are called unsaturated fats if one or more chains have unsaturated fatty acids. They are called polyunsaturated if they have many C bonds in their fatty acid chains. So, saturated fats have a regular structure. They pack tightly and form solids at room temperature. So, most animal fats are of this type. However, unsaturated fats have irregular shape. They do not really pack tightly and are liquid at room temperature. They are mostly oils. So, most plant fats are of this type. Now, what are phospholipids? Phospholipids are basically structural lipids. They form an integral part of the cell membranes inside and enclosing the cells. Structurally very similar to triglycerides. One fatty acid is replaced by a choline group. So, the choline group has both nitrogen and phosphorus in its structure. Now, the choline group is charged, which makes it polar. Okay, and it will hydrogen bond with water. That is why it is called water loving or hydrophilic. The rest of the molecule is non-polar and insoluble in water, which is, it will not form hydrogen bonds with water. It is hydrophobic or water fearing. So, because phospholipids have very two different tendencies towards water, what are they? They are called dash. What are they called? Does anybody know? We discussed in one of the previous classes. The phospholipid head group is hydrophilic, the phospholipid tails are hydrophobic. So they have two different tendencies towards water. That is why they are called, anybody? Sorry? Okay. Micelles are something different. I mean, you are going the right. No, not liposome. What liposomes are structures that that any kind of lipid can form. Micelles are also basically aggregate of surfactant lipid molecules dispersed in a liquid. But the term I'm looking for, they're not saying the term. These are substances that lipids can form. Bilayer is also something phospholipid can form. I'm saying phospholipid has a polar head group, which is why it is hydrophilic head group, hydrophobic tail. So they have two different tendencies. So what are they called? What is phospholipid called? What is another name for phospholipid? Micelles, bilayer, ribosomes, these are all things that that lipids can form. Sorry? I am talking about the characteristic. What is the term that is used to describe phospholipids? They are we cannot say that they're totally hydrophilic. We cannot say that they're totally hydrophobic. So what are they called? The characteristic term. Hydrophilic is water loving, hydrophobic is water fearing. So they are not totally hydrophobic, not totally hydrophilic. One part is hydrophilic, one part is hydrophobic. So what can phospholipids be on? The term that I am looking for is amphipathic. Phospholipids are amphipathic molecules. This means that they have a hydrophilic polar phosphate head and two hydrophobic fatty acid tails. Okay, these components of the phospholipids cause them to orient themselves. Okay, that's what I was meaning. So why are phospholipids amphipathic? Because they have hydrophobic tails and hydrophilic heads. So the tails of the tails will obviously hydrophobic. So an amphipathic molecule is something that has both, there is both the characteristics. Okay, micelle is obviously lipid molecules that arrange themselves in spherical form in aqueous solutions can be called micelles, but or micelles, so whatever the pronunciation is, micellar solution, micelles. Amphipathic is basically describing a molecule such as a detergent, for example, which has both hydrophobic and hydrophilic groups. So a phospholipid is a molecule with two fatty acids and a modified phosphate group attached to a glycerol backbone, right? So the phosphate may be modified by the addition of charged or polar chemical groups. So two chemical groups that may modify the phosphate can be choline or serine. Okay, so both choline and serine attached to the phosphate group at the position labeled R and via the hydroxyl group. So phospholipids are major components of the plasma membrane, the outermost layer of animal cells. Like fats, they are composed of fatty acid chains attached to a glycerol backbone. And unlike triglycerides, which have three fatty acids, phospholipids have two fatty acids that help form a diacylglycerol. Okay, so the third carbon of the glycerol backbone is also occupied by a modified phosphate group. However, just a phosphate group attached to a diacylglycerol does not qualify as a phospholipid. This would be considered as a phosphatidate, which is a precursor of phospholipid. Okay, phosphatidyl and phosphatidyl are very important examples of phospholipids that are found in plasma membrane. So phospholipid is called an amphipathic molecule because it has both hydrophobic and hydrophilic component. So single phospholipid molecule will have a phosphate group on one end called the head and the two side by side chains of fatty acids that make up the lipid tails. So the phosphate group is negatively charged, making the head polar and hydrophilic or water loving. Phosphate heads are thus attracted to the water molecules in the environment. The lipid tails on the other hand are uncharged, non-polar and hydrophobic or water fearing. So hydrophobic molecule repels and is repelled by water. Okay. So some lipid tails consist of saturated fatty acids and some contain unsaturated fatty acids. So this combination can add to the fluidity of the tails that are constantly in motion. Okay. That is all about phospholipids. Okay. So the choline end of the molecule is called the head and the fatty acid chains are called the tails. So the phospholipids will have hydrophilic heads and hydrophobic tails. So in water, the phospholipids will spontaneously form micelles or bilayers with their head. So this is what is called a micelle, with their basically head pointing outside towards the water because they are hydrophilic, and their tails associating with each other and avoiding water because the tails are hydrophobic. Okay. Now, what are steroids or sterols? These are basically, they have a much more complex structure than other lipids and a greater diversity of functions. Cholesterol is an important part of cell membranes. Vitamin D is necessary for some biochemical reactions to occur properly. Deficiencies can result in skeletal abnormalities, and other steroids and hormones, the sex hormones, testosterone, estrogen, and progesterone, these are all steroids. Now, what are proteins? We also discussed all of these before. The proteins are the most functionally diverse class of biological molecules. Protein diversity is the basis of the diversity of life. So, everything that organisms are composed of, all the parts, is made up of or by proteins. So, proteins serve as enzymes for defense, support, transport, motion, regulation, storage, etc. Okay, and proteins have a gross caloric value of 5.7 kilocalorie per gram, but the metabolism of protein creates toxic nitrogenous waste that must be processed for excretion from the body. Okay, and because of the toxicity of the byproducts of protein metabolism, proteins are not a good food source for providing energy. Proteins are necessary foodstuff for building new proteins. So therefore, energy purpose, it's the carbohydrates only. Okay, proteins are not really good food source for providing energy because their byproducts are very toxic. However, proteins do a lot of other functions, catalysis or transport, and a lot of other functions that we need to list. That's why proteins are necessary for foodstuff for building new proteins. So if you see, proteins are such a diverse have such diverse functions, have such diverse shapes and structures and all of that. Proteins can act as enzyme, help in catalysis. Proteins can help in defense, bodily defense like immunoglobulins, toxins, cell surface antigens, etc. Proteins help in transport, circulating transporters, membrane transporters. Proteins can help in giving support to the system. Proteins help in the motion. Proteins help in regulation, osmotic proteins, gene regulators, hormones, etc. Proteins also help in storage. Proteins are polymers of amino acids. So there are 20 different amino acids used in proteins. All have similar structure. All have a central carbon, an amino group, and a carboxyl group. The 20 different amino acids have different R groups. We have talked about proteins being polymers of amino acids joined through peptide bonds. Okay, so see, this is a peptide bond between these two amino acids. They come together, H2 is released, and polypeptide chain is formed. Now, all proteins are polypeptides. Not all polypeptides are proteins. Okay, a protein is a polypeptide that performs a function. So, if there is a polypeptide that doesn't really fold upon each other or is not really of that huge scale, it might not be called a protein. It will still be called a polypeptide. All proteins are definitely polypeptides. Okay, so there are so many different amino acids, but they have the same amino group and carboxyl group. So what gives them such different properties? So it's a variation of the side chain group or the R group that gives amino acids their different properties. They can be hydrophilic, hydrophobic, aromatic, non-aromatic, etc. etc. Okay, so phenylalanine, tyrosine, and tryptophan are aromatic amino acids. Okay. Now, the diversity of protein function results from a diversity of protein structure, which is result of variation in the sequence of amino acids. So, functional proteins can vary in length from about 50 amino acids to several thousand amino acids. Okay. A protein structure can be classified at several different levels: primary, secondary, tertiary, and quaternary. Primary structure is the amino acid sequence. If you can see, methionine, glycine, histidine, tryptophan, lysine. Secondary structure will be the regular coiling and the bending and the basically folding of the polypeptide that results from hydrogen bonding of amino and carboxyl groups in different amino acids. So these can form basically two characteristic, most important relevant characteristic structures: the alpha helix and the beta pleated sheet. Excuse me. The alpha helix and the beta pleated sheet. There are other structures, secondary structures that can also form, like oils, turns, and loops, etc. Now, R groups of different amino acids can interact in a variety of ways to produce more complex structure. Whereas hydrogen bonds are weak interactions between polar R groups. Non-polar R groups can have a weak attractive interaction. Okay. Now, coming to the tertiary structure, it is basically the bending of the polypeptide in 3D space due to the distant amino acids. The interaction can be covalent bonds such as disulfide linkages, hydrogen bonding, hydrophobic interactions, etc. And the quaternary structure is basically, one second. And the quaternary structure is basically the interaction of different polypeptides. Okay, the quaternary structure is the result of the same interactions as above. Okay, but not all proteins have quaternary structure. What do we mean by this? Can anybody try to answer? Can anybody try to answer what do we mean by this? Not all proteins have quaternary structure. So tertiary structure will be because of the bending and the folding of the polypeptide in 3D space due to the interactions of the R groups. Okay, the quaternary structure will be the interaction of different polypeptides. Okay, it's the same interaction as above. So there might be some proteins which which is only one subunit protein. Okay, so those proteins might not have a quaternary structure. So during the protein folding, there are some proteins that have different subunits. Okay, different polypeptide chains making up different subunits. Okay, so in that case, you might have a quaternary structure. Okay, but if that is not so, then in contrast to the first three levels of protein structure, not all proteins will have a quaternary structure, because some proteins can function as single units. They do not need other units for their function. Okay, so protein quaternary structure basically refers to biomolecular complexes of proteins with nucleic acids and other cofactors as well. Okay, so all proteins definitely have a primary, a secondary, and a tertiary level of structure. Okay, definitely, but quaternary structure is only formed. Quaternary structure is only formed only arises when a protein is made up of two or more subunits. So not all proteins will have quaternary structure. Okay. Now there are motifs and domains. So motifs and domains are basically components of structure within the protein only that will have different functions. Wait a second. One second. Okay, so motifs are basically super secondary structures that are basically channels and grooves formed by associations of pleats and helices. Domains will be regions of local tertiary structure along the length of a protein that can have different functions. Okay, so protein structural diversity is responsible for the functional diversity of proteins. Protein shape will cause protein function. Can anybody say why? Why is protein shape necessary for protein function? Anybody? So if the proteins, okay, so when you talk about protein, only the primary sequence is not important. That is definitely important. But the secondary, tertiary, all these levels of structures are equally important, because the level of these level of structures will basically dictate the folding of the protein. And the different folding of the protein will dictate whether the protein will be functional for what it is supposed to do, will be able to do its job, or is it non-functional, that is, it will not be able to do its job. So the protein shape will basically cause the protein function. So there are small changes in shape that can influence function. Proteins are also sensitive to environmental changes like change in pH, temperature, ion concentration, and all as a change in the shape of the protein and influence its functional properties. Like higher temperatures can result in weakening of hydrogen bonds, whereas proteins tend to denature or unfold at higher temperatures. As low temperatures strengthen hydrogen bonds, proteins become rigid at low temperature. Now, hemoglobin is a protein. Example, hemoglobin is a protein that carries oxygen in blood. So, hemoglobin has a quaternary structure. It has 2 alpha chains and 2 beta chains, and has an association with an iron containing molecule called heme. Now, hemoglobin is sensitive to a change in pH. Okay, it carries oxygen as well as high pH and does not at low pH. Okay, actively metabolizing tissues lower the pH of the blood as it passes through them. Okay, and what happens? Is that at the lungs the pH increases as CO2 leaves the bloodstream. Hemoglobin then picks up oxygen at the lungs and dumps it actively at the metabolizing tissues. Okay, that is what the role of hemoglobin is. So, had the structure of hemoglobin not been proper, not been in the way it is supposed to be, it will not be able to carry out its function. Okay. For nucleic acids, we have already talked about them being depositories and carriers of information. All the information needed to make living things work will be contained in nucleic acids, and they also transmit the information needed to make living things work. They are the basis of protein diversity and biological diversity. The information contained in the DNA makes organisms what they are. And obviously, there are two types of nucleic acids: DNA deoxyribonucleic acid and RNA ribonucleic acid. Both are polymers of nucleotides. Okay. So DNA and RNA are both polymers of nucleotides. They have a central 5-carbon sugar, a pentose sugar, a nitrogenous base, and a phosphate group. The sugar differs between the DNA and RNA. RNA uses ribose, DNA uses deoxyribose, and the two sugars differ in the functional group at the 2 position. If you can see over here, this is the position where the sugar is different. It is OH in RNA and DNA. Okay, nitrogenous bases found in DNA are adenine, thymine, guanine, and cytosine. The nitrogenous bases found in RNA are adenine, uracil, guanine, and cytosine. So thymine is present in DNA, uracil is present in RNA. Now the linkage between nucleotides is called phosphodiester bond. The sugars are pointed from the 3 carbon. The entire polymer will have a 3' to 5' orientation. One second. I don't know why this happened. It will have a 3' to 5' orientation. Excuse me. Okay, and the bases will extend from the side of the polymer. If you can see, the bases have been extended from the side of the polymer. Now, both DNA and RNA are synthesized by adding new nucleotides to the 3' of the chain. Here, new nucleotides are added. RNA can be a single-strand polymer of nucleotides, where DNA is a duplex. Okay, the DNA is a double-stranded molecule. The two strands wrap around each other as a helix. So, the two strands of DNA run in opposite directions. They are called, can anybody try to fill the blank? When DNA has two strands that run in opposite directions, so what are they called? What are the two strands called? Anybody?
Anti-parallel.
Correct. They are anti-parallel. So the 5' to 3' and the other strand will be 3' to 5'. So correct. This will be anti-parallel. The two strands are held together by H bonds. So obviously there are different hydrogen bonds between the nitrogenous base in the opposite strands. So A will only form hydrogen bonds with T, and there will be 2 hydrogen bonds. However, G will form 3 hydrogen bonds with C. Okay. So what are the other uses or functions of nucleotides? They are also important in important energy transfer reactions like ATP. Okay, 1 second again. This is ATP. The bonds between the phosphate groups are high energy bonds. So the energy is captured in the synthesis of the phosphate bonds of ATP and released in the breakage of those bonds. Then we have nicotinamide adenine dinucleotide NAD, which is composed of 2 nucleotides as usual. And NAD is used in many oxidation reduction reactions to accept or donate high energy electrons. Now the other questions that we had was about stereoisomers, mirror images calling enantiomer, something like that. So if we try to talk about isomers basically having the same molecular formula but they are different in some way. Okay, so there are isomers. If they have the same connectivity, they are stereoisomers. If they don't have the same connectivity, they are called constitutional isomers. In stereoisomers also, if they are different by rotations about single bonds only, then they are called conformational isomers. Otherwise, they are called configurational isomers. And if they are mirror images, non-superimposable mirror images, then they are called enantiomers, non-superimposable mirror images. Whereas, if they are not mirror images, then they are called distereomers. So the question was this, which one? They are not called enantiomers, and the question asked which of this is true. So then that is false, because enantiomers are non-superimposable mirror images of each other. Okay, so basically if you talk about this part of isomers, other part will be diastereomers, but if you stick to enantiomers are stereoisomers that are non-superimposable mirror images. So a molecule with one chiral carbon can exist as 2 stereoisomers, okay, enantiomers. So enantiomers differ in the configuration, RS configuration at the stereogenic center. So enantiomers have identical chemical and physical properties in an achiral environment. Enantiomers rotate the direction of plane polarized light to equal but opposite angles and interact with other chiral molecules differently. Okay, so a molecule with 2 chiral carbons can exist as 3 or 4 stereoisomers. Distereomers are, however, stereoisomers that are not enantiomers. Okay, so that is what distereomers is all about. Now, enantiomers have identical chemical and physical properties in an achiral environment. However, distereomers have different chemical and physical properties such as melting rate, solubility, etc. Okay, so to convert between enantiomers, both chiral carbon atoms must change configuration. All stereocenters must be different from molecules to be enantiomers. However, to convert between diastereomers, only one chiral carbon atom must change configuration. At least one stereocenter must be different, and at least one stereocenter must be the same for molecules to be diastereomers. Okay, so that is what are mesocompounds. Just for general knowledge, I am saying mesocompounds are basically they have an even number of asymmetric atoms of opposite configuration, and they have an internal mirror plane bisecting the molecule. So each half of the mesocompound is a mirror image of the other half and cancels the optical activity of the other. Thus the molecule is achiral. Okay. So you see this, enantiomers are basically non-superimposable mirror images. See, these are mirror images, but they cannot be superimposed on each other. So molecules are non-superimposable if there is no orientation in which the terms of both the molecules can be superimposed. Okay. A little bit about chirality. A molecule is chiral if it has an enantiomer. That means it does not have mirror symmetry. A molecule is achiral if it does not have an enantiomer. That means it does have mirror symmetry. So we have talked about some of the terminologies before as well in stereochemistry. Stereocenter, which is an atom about which exchange of two groups produces a stereoisomer. We have a chiral center type of stereocenter, which is a tetrahedral atom that is bonded to four different groups. Okay. I have talked about the Fischer projection last day, which is basically a very convenient way of depicting complex molecules having more than one stereocenter. So the intersection of a horizontal line and a vertical line will indicate a carbon stereocenter, and the substituents on the horizontal bonds are understood to point towards you, okay? Whereas the substituents on the vertical bonds are understood to point away from you. About chirality and conformational isomers, rotation about single bonds can determine whether a molecule is chiral or achiral. If a molecule and its mirror image are rapidly interconverting conformational isomers, then the molecule is effectively chiral. Okay. We have talked about Howard projections before as well. Howard projections are helpful in determining the chirality of all cycles. So if we take the chair conformation of cyclohexane to a flat hexagon, it allows us to quickly and accurately determine whether it is chiral or achiral. Okay. Will not go into too much details. For constitutional isomers, due to different connectivities, these isomers must have different physical and chemical properties. For enantiomers, they have the same connectivities, okay, as you can see over here, and precisely the same polarity. So their physical and chemical properties are mostly similar. For diastereomers, they have the same connectivities but have different 3D arrangement. So they have different properties. So basically we have been talking in the terms of optical activity. So chiral molecules interact with plane polarized light. When all photons from a light source have their electric fields oscillating in the same plane, then the light is called plane polarized. And we have a polarizer. Basically most light sources emit light that is unpolarized. So a polarizer generates plane polarized light by filtering out light whose electric field oscillates in any other plane. So if plane polarized light passes through a sample of compound, then the plane in which the light is polarized can change depending upon whether the compound is achiral or chiral. Now enantiomerically pure chiral compounds are said to be optically active because they rotate the plane of polarization. Achiral compounds are said to be optically inactive because they leave the plane of polarization unchanged. Okay. Enantiomers rotate plane polarized light. So one enantiomer rotates plane polarized light in one direction, whereas the other enantiomer rotates it in the opposite direction. Enantiomers have identical physical and chemical properties except the direction at which they rotate polarized light. Okay. So you can see this is plane polarized light. The interaction of the light with this molecule rotates the plane, but interaction with the molecule in this mirror image position rotates the plane back. The rate rotation will be 0. Okay. So we have talked about all these things pretty much. So coming back to the question that we started with, let me go back to the question. Now this one. Yeah. So the question that we started with was, the biomolecules present in E. coli are different from those in humans. True. Obviously, E. coli is a prokaryotic system, human is an eukaryotic system. So obviously the biomolecules present in both these organisms are very different from each other. Okay. The role of nucleic acids is to act as reserve energy sources for the cell. This is not true because the role of a nucleic acid is basically to transfer genetic information from one generation to the next. Stereoisomers that are not mirror images are enantiomers. This is false because stereoisomers that are non-superimposable mirror images are enantiomers. This statement is false. The huge diversity of living organisms are created from relatively fewer metabolites. Again, true. Okay, because obviously we have a huge diversity of living organisms. There are so many different kinds of living organisms with different habitats, staying at different altitudes and all of these, but they are created from relatively very fewer metabolites. We have certain elements, carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and using these only there are very few metabolites that can be prepared, that can be synthesized in the system, that are in the system, and the huge diversity of living organisms are created from a relatively fewer metabolites. The statement is true. Then glucose has a 6 carbon molecule. Glucose has 6 carbon molecules and is a ketose. That is false because glucose does have 6 carbon molecules, but it is an aldehyde, aldose. Okay, so it is not a ketone. Fructose is a ketose. So the last statement is also false. So the correct statements in this question will be the biomolecules present in E. coli are different from those present in humans, and the huge diversity of living organisms are created from relatively fewer metabolites. These two are the correct statements. Okay. Anybody has any question regarding this? This was the last question for today. If anybody has any question, you can let me know. You can type in the chat box or you can just unmute and let me know. We'll try to solve this. Okay, so if nobody has any questions, maybe we can conclude the session. We'll meet again next Monday. I'll stop sharing my screen. So these videos will be, session recordings will be uploaded as videos on YouTube. Okay, so I'm guessing nobody has any question. Then maybe we can stop this session and we'll meet again next Monday. Thank you. Thank you.